right? IAPC 14 from Brazil, right? Okay,
Yeah,
thank you.
correct.
Good morning. This is Hong Kong time in the morning. Okay, so I say good morning to you. Welcome to the Global Youth Science and Technology Bowl 2021. I'm Haley, the MC of this judging panel. This is team from Brazil. Their project title is about periodic table. Okay. First, we would like to thank you for attending the judging online today and for your effort made in the past few months for developing the project. Today, we are glad to have Professor Edwin Chen from the Chinese University of Hong Kong and Professor Zhang Xiujin from City University of Hong Kong to be the judges of this competition. Please note that my window will turn into a countdown timer when the judging officially starts. You will have 5 minutes to present your project with screen sharing of PowerPoint or any other materials. We will remind you the time after 5 minutes, and you have to stop the presentation when the time's up, okay? And there will be 4 minutes for the Q&A section after your presentation. Do you have any questions?
No, it's fine.
Okay, that's
Thank
great.
you.
So you may share your presentation materials now. As I know you got a PowerPoint, you got a PDF.
Yeah, I'm starting to share it.
Okay, that's great. Okay, so let me set the countdown timer for you. Okay, ready? You may start your presentation now.
Okay.
Thank you.
Hi, my name is Victoria, and this is my colleague Leticia. And today we'll be presenting our project, a periodic table, a scientific, inclusive, and technological development instrument. Our project started in 2019 due to the celebration of the periodic table's 150th birthday. In addition to that, in 2024, inclusive education becomes mandatory in Brazilian schools. So we built a 3D periodic table, sustainable, interactive, and technological.
So the project's goals are to demonstrate the importance of inclusive actions at school and in society as a whole, making the teaching and learning of chemistry and natural science more attractive through the use of interactivity, and also use technology to learn and spread knowledge, as well as make the material accessible to the biggest number of students as possible. So talking about the interactivity, each element is represented by a cube, like this one I have in my hands right now. And the colors symbolize the different groups of the table. And each cube has various information about a certain element. So chemical informations, curiosities, applications, the space that you can find in the common periodic tables, and also 2 other faces that are related to the other parts of our project. So we'll be explaining later. So by this, you can find a possibility of a better learning about the subject. And also in normal times, all the students from our school have access to the periodic table to study and to use it as a tool. Unfortunately, because of the pandemic, it becomes a little harder, but in normal times, they can access it at any time they want. Now about sustainability, we use sustainable material to build the table by reusing cardboard boxes to make the cubes. And besides cardboard is a light material, it is cheap and easy to work on. And thinking about the durability of the cubes, we developed new ones made with reforested wood. And by reutilizing the cardboard and using reforested wood, we're fulfilling the 17 Sustainable Development Goals from the United Nations.
So, when it comes to the inclusive part of the project, we insert the resources for the visually and heard impaired people through the abbreviation in Libras, as my colleague is going to show for you guys. Libras, the Brazilian language of the signs of the element, as well the signs in Braille, the rhythm system for the visually impaired people, and a texture that diversifies the different groups of the table. Thinking about the moderate disabled people, initially we developed a type of claw, so they could reach the highest elements of the table, since that is 2 meters high, by themselves in autonomous ways. However, aiming to improve our project and make it accessible to all people, we are currently planning to insert a mechanical claw to the table, a matter that we will explain in the technology part.
So talking about technology, each cube has a QR code like this one. And when this QR code is scanned, it directs the user to a website that we developed by ourselves. So you can see a picture here in the slideshow. It's a screenshot from our website. And in the website, you can find all the information you have in the cubes about elements and much more, and also audiobooks in English and in Portuguese. Besides that, we're trying to develop an artificial intelligence system with tags of electronic identification in the reforested woods. So the user will be able to select an element by a self-service terminal and will be able to choose between hearing an audiobook about that element or ask a mechanical claw to grab and deliver the cube in the user's hands.
So it is worth mentioning that because of the pandemic situation and the distance learning, our website has gained fundamental importance. And besides that, we can notice a change in the body of in the students' body of our school. Thank you so much for watching, and my colleague is going to
Samara. So it's important to say that the project is still in development. So the website is being concluded as well as the artificial intelligence system. And we also are open and appreciate suggestions from you guys. So if you have anything to tell us, and that's basically this. Thank you so much.
Hey, thank you. That's the end of your presentation, right?
Yeah.
Okay,
yeah.
Thanks a lot. Okay, now we have time for judges to ask you some questions. Okay, 4 minutes for this session. Any question from judges?
Okay, maybe I start first. It's a very interesting tool, right, for people to learn the periodic table, and it contains different kinds of facts, right? But I would like to know to what extent your AI system has developed so far, the artificial intelligence system.
We are working with another school to implement it. So because of the pandemic, it had been delayed. We started the project in 2019, like my colleague said, and this idea came up in the end of 2019. So we got in vacation, and when we come back to school and started to think about working on that, we had to stop everything because of the pandemic. And because of that, we couldn't try to finish and go after it, and also because the school is in another city, so it's really hard to make a physical contact with them, and it is a lot easier when we can talk in person and stuff, and this is something we cannot do right now. So we are still projecting it and doing this kind of things, but for now the table is available in our school in this form that you can see in the picture in the slideshow.
Okay, thank you. Another one you're talking about, you have the Brightly, that means the reading system for visually impaired people, right? But yeah. I mean, the information contained, is it equivalent to the information available to the normal people? Because I just see one side, you have a few words right in Brightly, but I don't know whether it contains most of the information that normal people can get access to.
So we just implanted this little sign right here because in our school, we don't have any people that are visually impaired. So our main goal with this tool was to make people want to learn more about that, because to implant the whole signs in the whole cube would be very expensive, and it's really hard to find people that do this kind of things. So our main goal in planning it was to make people like it and want to learn about it. And it worked very well because people started to interact with the table and try to reproduce the signs in Libras and put their fingers in the Braille and try to identify it. But for the visually impaired people, we prepared something in our website so they can access it and hear the audiobooks that are also available in English.
Okay, thank you. Professor Chan, please.
Thank you, Professor Chang. So very, very impressive work.
Thank you.
I'm amazed that periodic table can be presented in this way, and I'm sure that all the school students in your school must have a good time. Well, one question I have in mind is about your design. So can you tell me, okay, of course, you have realized the design and also executed an education program based on the periodic table you built. But what was the original idea of building such a table? Do you want to, for example, educate students in terms of the knowledge of the periodic table behind, or you would like to promote inclusiveness and also promote the sustainable development goals? I'm a little bit not very clear about this.
And I don't know if my friend can reply, but since we have just a few time, it's kind of a mixture of both. So the table has wheels, so the teacher can take it to the class, and it facilitates a lot to understand the content since you can see it, you can grab it, and also these other things you mentioned. The time's up. I wish I could speak more.
So please complete, finish it. Yeah, it's okay.
Oh, okay. Yeah. So it was a mixture of everything. We wanted to include all these tools that are related to the basis of our project, like sustainability, interactivity, but also the main goal was to make it a tool that our friends and colleagues and people from other grades could use to learn the subject in a better way, especially get to high school and start to learn more about chemistry and these subjects related to that. It's very hard because you're not used to that. So by the table, the students could learn it in a better way since they could visualize it, they could grab the books, they could see the information right in their faces, not only in the normal way that we see in paper. It was way more like attractive, like we mentioned before, and also the colors, the different colors that stand out a lot.
I understand you. So one more follow-up question, a very quick one. I hope Haley will allow. So do you have an accompanied teaching program to go with this design? Otherwise, students would just go and explore the way as they want. They play around with the periodic table. Do you have a program behind?
You mean like the way school teaches?
Yeah, the teaching objectives and that.
Yeah, we do. In the first year of Brazilian high school, you learn the periodic table and you learn how to use these informations, the chemical informations we put right here. So that's why it's very useful for these students. I don't know if I answered your question.
So can I say, so in short, so it is a self-learning invention for students to understand periodic table. Is that correct? Okay, thank you very much.
Thank you so much.
Thank you.
Okay, thank you.
Thank you.
Thank you, team. Okay, and you may leave the meeting room now. See you tomorrow again in the activities and our ceremony. Okay, take care.
Okay, bye-bye.
Okay, goodbye. Thank you.
Thank you. C19, right?
That's correct.
Hello, welcome to Global Youth Science and Technology Bowl 2021. I am Haley, the MC of this judging panel. This is Tim from Mexico. First, we would like to thank you for attending the online judging today and for your effort made in the past few months for developing the project. Today, we are glad to have Professor Evan Chen from the Chinese University of Hong Kong and Professor Zhang Xiujin from City University of Hong Kong to be judges of the competition. Please note that my window will turn into a countdown timer when the judging officially starts. You will have 5 minutes to present your project with screen sharing of PowerPoint. We will remind you the time after 5 minutes, and you will have to stop the presentation when time's up. And there will be 4 minutes for the Q&A session after your presentation. Do you have any questions?
No.
That's great. Please share your presentation materials now. And I'm going to set the countdown timer for you.
Thank you.
So, are you ready?
Yes.
Okay, please start your presentation now.
1 out of 3 people globally do not find themselves within reach to save drinking water. Mexico is in desperate need for a solution, as over 80% of the residual waters produced ought to not be treated, meaning that more than 22 billion liters of water available for human consumption are wasted daily. Statistically, this quantity would be enough to satisfy Hong Kong's total population water needs for 22 days. That being said, the question is, why are we avoiding solving this problematic? Will humanity wait until the hydro crisis is no longer reversible? We are Bernardo Landa, Regina Sanchez, and Jose Pablo Castelazo. Welcome to our presentation. Use of Puntia ficus indica as a coagulant for wastewater treatment. The investigation we carried out in order to achieve a solution for this and to determine whether a natural coagulant could be as efficient as a chemical one. Likewise, we created the quadruple E proposal, which consists of evaluating 4 aspects of the coagulants, those being the environmental compatibility, its ethical responsibility, the economic benefits for its users, and the efficacy. After researching, the nopal mucilage, which is a mixture of polysaccharides and a fibrous substance, was selected. We chose the nopal, prickly pear pads, which are a Mexican endemic type of cacti that is seen as one of the major blocks in our national cuisine, in addition to being used for traditional medicine purposes.
The objectives consist of contrasting both coagulants to identify the most eco-friendly one, the identification of pollutants in the aluminum sulfate samples, and determining the possibility of mass-producing nopal mucilage.
The hypothesis proposes that using nopal mucilage during the coagulation process for wastewater treatment will result in the turbidity decreasing and the other parameters being similar or better in the samples, in contrast to a chemical coagulant, thus forming an easily treatable sludge. The methodology was divided into 2 sections. The first one consisted in the mucilage extraction from a nopal cloud. After being cleansed, the tissue was macerated in purified water for a day at 16 degrees Celsius, while subsequently being stored in a refrigerator. Once done, the first filtration occurred. Following that, the supernatant was subjected to a 75 degrees Celsius water bath for a day. This led to the mucilage precipitation from the aqueous phase being carried out by adding ethanol in a 1:3 ratio. Lastly, the mucilage precipitate was obtained from a second filtration. Then, after a 5-liter water recollection from the Cuemanco Canal in Mexico City, measurements for the pH, dissolved oxygen, electrical conductivity, and turbidity were performed with diverse sensors. In sequence, 3 samples were treated with 10 milliliters per liter of aluminum sulfate prepared at 10% and 3 with 0.5 grams of mucilage. Always stirred until flock formation occurred. Accordingly, their measurements were taken.
Regarding the results, their permitted parameters were considered according to the Mexican and Latin American standards. As it can be seen, these were classified depending on whether they achieved or failed these parameters. Satisfactorily, the mucilage was the most efficient. By contrasting the results, the mucilage pH came out perfectly, while the aluminum sulfates reduced drastically. Then, the oxygen levels presented a low result for the mucilage. However, it is expected to improve by adding more of it to the samples. All conductivity levels are inside the standards. Yet, the results from the aluminum sulfate raised questions regarding its effectiveness and viability, especially due to its relation to Alzheimer in the elderly. Lastly, despite the mucilage turbidity not reaching the minimum requirements, enlarging its quantity should permit it.
Comparing them, the natural coagulant was more beneficial. This was not only due to being around 39% cheaper, but also because of its beneficial effects on the human's health and the capacity of purifying 2,000 liters of water with just 1 kilogram of mucilage. Finally, the limitations seen with the lack of financial capacity, the COVID-19 pandemic disruption in the trialing, and some uncertainty about the adequate quantities of mucilage needed.
Aside from that, some conclusions can sum up the experiment. The hypothesis was partially accepted due to the mucilage turbidity result, while the main objective was successful. Then, due to the limitations, the presence of pollutants in the aluminum sulfate samples was assumed, but not confirmed. The possibility of mass producing the mucilage was demonstrated, thus meaning the experiment overall result was extremely successful. Finally, the employment of noble mucilage represents a circular economy process beneficial for Mexican rural communities. As for further investigation, a trial expansion to determine the amount of mucilage needed for better results will be made.
So, what do you say? This problem can be solved, and we have the solution. It is called no-pole mucilage. Thank you for your time. Here are the references consulted.
Thank you for your presentation. So, now we have four minutes for judges to test to ask you some questions. Okay.
Let me start first. So it's a very interesting investigation project. So I just want to have some background question first. So do you think the cactus material used is already the best, and why?
In our opinion, the no-pole cactus, the prickly pear pods, are the best cacti available, because we can do this experiment with other types of cacti. However, the results and availability are not the same. In Mexico, thousands or even millions of tons are produced in Mexico, well, yearly from no-pole. So that is why.
And for a question, so how do you identify the species identity of your cactus used every time when you do the experiment or do the extraction?
So we did not have that specifically planned in our objectives, but seeing that it is a really common species in Mexico, it is somewhat easy to find them. However, by being able to carry out the same experiment with other types of cacti, we could do it by not identifying these types of no-pole.
Okay. One last question is, I noticed that in some of your data presented in the table, you have an error bar. So can you tell me the sample number or the number of experiments you have repeated for the experiments that you presented in the table?
Yeah, as we said, we made 3 samples for aluminum sulfate and 3 samples for the natural coagulants. So the error bar consists of those 3 samples.
And we weren't able to make more samples because of the COVID-19 pandemic disruptions. We had 3 phases for the experiment. The phase 1 was to see if the no-pole mucilage was a good coagulant. However, the second phase was to correct some mistakes and some information that we were able to improve. And the third phase was to mass produce it and to contact some other providers in order to implement this strategy into the Mexican community. However, due to the pandemic limitations in our country, we were not able to perform that, but we have that as a plan to do for further investigation.
Okay, thank you.
Okay, maybe I ask a more basic question about the mechanism. So you use the mucilage, right, in treating this wastewater. So what is the mechanism behind that helps to treat the sewage? That means the mechanism of sewage, how it can clean up the water.
Well, our mechanisms consist of coagulation, flocculation. So the mucilage and the aluminum sulfate, they charge the particles, so they attract each other, and they sedimentate, so we can separate them easier.
Okay.
Yes, and we have a very thorough methodology in which we made various steps to try to treat this sewage water, but it was difficult because the Xochimilco's water is really polluted, so it was a tough task.
But why I ask this question is, you know, the mechanism. Actually, later on you can find replacement, right? Using the same principle, but of course you can homemake or make it in a mass production in the industry, right? Using synthetic materials, using the same mechanism. Another question is about the cactus. Does this species grow very fast?
Yes, it actually does. It is one of the the fastest grown species in Mexico, and in the world. It grows from 0 to 6 months, and at 6 months it is considered to be mature enough to be consumed and to be used for this. It can live up to 2 to 3 years, and it is really, really common in this country.
Okay. Just one last question. I'm sorry. I would like to know whether can it be recycled? Can this mucilage be recycled so that you can treat the sewage more than once using the same batch of mucilage?
Well, the mucilage, we didn't really focus on that, but the residues of the nopal can be reused and can be eaten, so no waste is generated.
And
but
it
we
might
didn't
be
really focus on the recycling of the mucilage.
And it might be reused, but we have to make the same procedure once again with the remains from the water treated. So we will have to be able to try to do further investigation in order to analyze it. Probably its efficacy would lower a little bit because of the percentage of mucilage used for each time, but it will probably work properly.
Okay, thank you.
Okay, thank you, judges, thank you, team. And you may now leave the meeting room and see you tomorrow again in the activities and award ceremony. Take care, bye-bye.
Thank you very much. Bye.
Hello.
Yes, I can hear you.
Yeah, thanks a lot. Welcome to Global Youth Science and Technology Bowl 2021. I'm Haley, the MC of this judging panel. This is Tim from South Korea.
Yep,
yes, first, we would like to thank you for attending the online judging today and for your effort made in the past few months for developing the project. Today, we are glad to have two judges, including Professor Evan Chen from the Chinese University of Hong Kong and Professor Zheng Xiu Jin, City University of Hong Kong, to be the judges of the competition. Please note that my window will turn into a countdown timer when the judging officially starts. You will have 5 minutes to present your project when screen sharing of PowerPoint, with screen sharing of PowerPoint or any materials. We will remind you when the time the time after 5 minutes, and you have to stop the presentation. And there will be 4 minutes for the Q&A session after your presentation. Do you have any questions?
No, it starts from when I share the screen, right?
Please share your presentation materials now.
Okay.
So I'm setting the countdown timer, and you may start the presentation now.
Okay. Hi, my name is Dong Kim, who is an 11th grader in South Korea. I have made an experiment about the effects of food ingredients and smoking on antibiotic resistance of S. mutans bacteria. This is my research motive. It's to find the harmful effects of antibiotic-resistant bacteria on human health, and to know more about the major pathway to infection in the body's various channels, and to know more exactly about Streptococcus mutans, which is also known as the cavity-causing bacterium, which will be introduced later, and then to know more about the effects of smoking and various food ingredients on antibiotic resistance. Here are the purposes of my research. I have total 4 of my purpose, which is my first purpose was about the collection of the samples of S. mutans bacteria from our surroundings, and examining between smokers and non-smokers, and then comparing and contrast between people who take in sugar and people who don't. And to compare between people who intake xylitol while there's people who don't. So our main ingredient over here in this experiment is the Streptococcus mutans bacteria, which decomposes sugar from the food remaining on the teeth, which will make acids, then melts the enamel layer of the teeth, causing cavities. We also need the GYSTB growth medium to cultivate the S mutants. Any of these antibiotics can be used, fulgrams, avofloxacin, lincocillin, or ammonelic. It's to increase the possibility of the resistance and by increasing the exposure time. We also need xylitol in order to contrast and compare after later of the experiment. So here are the samples of S. mutants from our surroundings. I got a total of 8 samples, and then diluted to 1/10 to 1/1000 concentration, and then cultivated them at 36 degrees for 2 to 3 days. As you can see, these yellow particles are the S. mutans bacteria colonies. And over here, the river water has almost no S. mutants, while the subject B saliva has lots of S. mutants. So this is the proportion of the antibiotic-resistant bacteria of sampled S. mutants from our surroundings. It's been researched that the general antibiotic-resistant bacteria are like 10% of the whole entire bacteria family, which made us to go through a process of diluting a concentration 10 times stronger than the usual dilution factor, or else the results are going to be a little off or mistaken. So as shown in the results, the S. mutans bacteria were not commonly found in the sources other than the human oral cavity, such as water or soil natural conditions. And then by this, it can be predicted that more cavities in the mouth, the more S. mutans bacteria can be found. So our first comparison was comparing between the people who smoked and people who didn't. In this experiment, we could see that the non-smokers has almost no S. mutants found, while the people who smoked had lots of S. mutants, meaning that there's going to be more S. mutants in the oral cavity. And the antibiotics that we used in this experiment was called cefaclor. And our next comparison was between the sugar intake. When we tested between the sugar intake, we tested by giving them a candy full of sugar, and then we gave them for an hour, and then we replaced it after 10 minutes. And then when we checked it, it really had no difference between people who took in sugar and people who didn't. The S. mutans population really did not change, meaning that since there was no significant change, sugar intake had no effect on antibiotic resistance. Our last comparison was the xylitol intake, which was our main. We also gave them the gum to chew, and then we also replaced them with a 10-minute term. And then when we gave the xylitol, when we examined the saliva after they chew it, they had a very interesting result. Based on the result, the number of colonies was reduced by no less than 30%, and even more than 80% in severe cases after the xylitol intake. Which means it's interesting that the number of the antibiotic-resistant S. mutans bacteria has really increased against certain antibiotics after xylitol intake. which the antibiotics, again, is a cephalchlor. To sum up my experiment, like in conclusion, if there's more cavities in their mouth, of course, there's more S mutants found since it's found in the oral cavity. And then by our comparison experiment, which is the smoker and the xylitol intake, it can be seen that the S mutants evolved to increase antibiotic resistance in response to conditions that pose a threat to their survival. So basically, even though their population decreases, they increase their antibiotic resistance by increasing their survival. Therefore, further examination, I think, is needed to various factors that may affect the antibiotic resistance in numerous bacteria present in the mouth, like S mutants. Thank you.
Thank you. Now is Q&A session. Judges, please ask. Professor Chen.
So let me start first, maybe this time.
Okay.
Interesting investigation, and I would more like to ask a question about your sample collection.
Oh yeah, sample
the
collection.
rationale behind. So in particular, the sugar condition. So why you picked 1 hour as the as a time for the subject to take in candy? So why is this time point?
Oh, so why did I take sugar as my ingredient? So I thought, so after I found that the S mutants are inside our oral cavity, like over in our mouth, by looking at slide 6, I gave the surroundings, and they can be seen that inside the saliva there was a lot of S mutants, right? So I thought giving like a food ingredient, like maybe salt or sugar, but sugar would be a better choice than like other ingredients because it's easy to look at it since a lot of food has sugar inside. So if experimenting sugar, I thought we can know the basics of if the sugar affects the S mutants or not.
But the time duration is only 1 hour. So what do you think within that hour be happening to the S mutants?
Oh, so for an hour
faster or quicker or?
Oh, you mean why did I take the reason, like what's the reason I did only for
1 hour after the sugar intake, and then you collect the sample.
After I collected the sample of after an hour, I explained that there was no difference, right?
Yeah, so the question I asked was why 1 hour only? Why not 2 hours, 4 hours, or overnight?
That's because since like the take-in of the sugar, there was no difference at all. So I did this for exactly, I think, 3 hours and like about 2 times so that there's no mistakes. But there was no difference after an hour. So I set it up as a 1 hour since
thank you.
Everything really had no difference.
Thanks.
Okay, so what is the sample size? How many people were involved in this study? The smokers and non-smokers and those?
Okay. These are all 3 different people, all my family. And then they said, all right, let's try this because I told them about the experiment and they were also interested. So I tried to tell them since people use smoke, how about having the smoke? There was 3 people involved in total.
3 people. Do they involve in the same experiment?
No, they don't. They're all 3 different people. So that the saliva doesn't get mixed, even though like a day had passed or not. Because the results has to be very, very clear and organized so that there's no other factors.
Before you designed this experiment, is there any literature record telling about this kind of observations? Say whether there's any relationship between the antibiotic resistance and the consumption of xylitol or smoking?
I researched about the smoking, so I put that smoking in, but I thought sugar and xylitol was my idea of how about putting it in. So because I thought it would be interesting. Sugar is the same reason as before. Xylitol, because I chew a lot of gum, and then since S mutants are inside our oral cavity, I chose gum. Yep, I also chose gum. That's the reason why. Because I chew it a lot.
Okay, because the companies they sell xylitol claim that actually consuming xylitol should result in less cavities, right?
Yeah. I also know about that, but yeah, I tried it.
So that means your result seems probably contradictory, right? To that.
It's a little contradictory, but it's based on my results, so it can't be always right. But yeah, I'm following my result since I tested it myself.
Okay, thank you.
Thank you.
Thank you, team. Thank you, judges. Team, you may leave the meeting room now. See you tomorrow again in the activities and our ceremony. Take care.
All right, thank
Bye-bye.
you.
Bye-bye.
Bye.
Bye-bye.
morning.
Hello. Good morning. Good morning. You are from Singapore team, right?
Yes.
Do you have another? Do you have the third member?
Yeah, he's
yeah. Hi. Hello.
Hello.
Welcome to the Global Youth Science and Technology Bowl 2021. I'm Haley, the MC of this judging panel. This is Tim from Singapore. First, we would like to thank you for attending the judging today and for your effort made in the past few months for developing the project. Today, we are glad to have Professor Evelyn Chan from the Chinese University of Hong Kong and Professor Zhang Xiu Jin from City University of Hong Kong to be the judges of the competition. Please note that my window will turn into a countdown timer when the judging officially starts. You will have 5 minutes to present your project with sharing your PowerPoint, or you will remind the time after 5 minutes and you have to stop the presentation when time's up. And there will be 4 minutes for Q&A session after your presentation. Do you have any questions? Okay, no problem. Great. Now you can share your presentation material. And I'm going to set the countdown timer for you. So, team, are you ready to present? Okay, please start now.
Hi everyone, I'm Ray Ann.
I'm Irvin. And I'm Brian.
And today we'll be talking to you about the isolation and characterization of human proteins, the Interactivity protein of SARS-CoV-2. So why do you want to work with SARS-CoV-2? Well, millions of people have been infected by this virus and people from all over the world has been affected by it. And to give you more information about SARS-CoV-2, when the virus enters the human cell via endocytosis, it aims to evade the host immune system and continuously replicate its viral RNA. SARS-CoV-2's viral RNA is split into 10 open reading frames, RF1 to 10. Out of these open reading frames, the functions of RF10 is the most uncertain since it is not found in SARS-CoV-1.
From a paper, severe cases of SARS-CoV-2 infected have been found to have higher levels of RF10 as compared to moderate cases. Since severe cases had higher levels of RF10, we suspect this could have led to a decreased immune response. Hence, we want to know more about it by finding potential interactions with RF10. So using protein-protein interactions in this case. Why? Well, from the principles of virology, viruses use their own proteins to target host proteins. Proteins also have a specific 3D shape, and if two were to interact, there must be a purpose behind it. So to answer, how does SARS-CoV-2 evade the host immune system using RF10? Well, we did several experiments using these two hybrid and split ubiquitin assays, and then we made plasmid constructs for both assays to experiment and find out potential interactions with RF10.
The main purpose of our assay is used often as bait to catch human proteins that interact with it, allowing us to better understand this virus. After screening through hundreds of thousands of colonies, we finally found yeast colonies that interact with that grow on his minus plates. We had to make our own ORF10 gene and inserted this gene into a yeast hyperplasmid and confirmed it using sequencing. We obtained our results through sample cell dilution in yeast hybrid system and found two synthetic peptides, Y10B15B and Centin-A, which were found to interact with ORF10. So what is yeast hybrid and how do we use it? We binded ORF10 to the GAL4 DNA binding domain and our protein from our library to the GAL4 activation domain. When ORF10 and the protein from our library interact, the GAL4 DNA binding domain and activation domain will come in close contact, allowing the history reporter gene to be transcribed. Hence, our yeast cells can make their own histidine, allowing them to grow.
As can be seen in the orange box, there's cell growth in lanes 2 and 3 on his minus plates. This tells us that the cell is able to make its own histidine, thus showing us that Y10B15B and Y10B17A can interact with RF10.
After more screening, we found another interactor of RF10 called beta-2 microglobulin, also known as B2M. From literature, we found that B2M is split into two regions, amino acid 1 to 20, which is a signal peptide, and amino acid 21 to 118, which is a motor peptide. To check which region of B2M RF10 interacts with, we perform split ubiquitin assay. But to do this, we first split ubiquitin into its CUB and NUB halves, and tagged RF10 to CUB and RR3P, while B2M is tagged to NUB. If RF10 were to interact with B2M, the two halves of ubiquitin are brought together. UBPS will recognize this as a whole ubiquitin molecule cleaving off RR3P. This will allow cells to grow on plates containing FOA, fluorouric acid, since RR3P is not present to convert FOA into toxic fluorouracil. So, from the purple box, you can see cell growth on lane 2 instead of lane 3 on plates containing FOA. This proves that RF10 only interacts with the signal peptide of B2M, but not the motor peptide.
So B2M is part of MHC class 1, which is responsible for alerting the host immune system of a viral infection. We hypothesize that when ORF10 interacts with the B2M signal peptide, it causes a degradation of B2M, thus preventing MHC class 1 from presenting viral antigens, decreasing the immune response. So to confirm our protein-protein interactions in vitro, we had to ensure our proteins were soluble first. Western blot was used to detect for soluble proteins. Looking at the orange box, ORF10 is not soluble when tagged to GST, but was soluble when tagged to MBP, as can be seen from the blue box. Moving on, looking at the green box, B2M tagged to GST is slightly soluble when prepared with buffers PBS and TBST. In future, we hope to confirm that ORF10 and the B2M interaction can still occur in vitro using a pull-down assay, using the samples we have prepared.
We would also like to find out if the degradation of B2M can still occur in tissue culture cells when the ORF10 gene is expressed.
Lastly, can the synthetic peptides Y10B, 15B, and Sentin-A interfere the interaction between ORF10 and B2M, possibly allowing the immune system to be alerted of a foreign infection? And if so, can we use some sort of drug for patients infected with SARS-CoV-2? We really do hope that our research will be able to help fellow researchers and scientists better understand the inner workings and mechanisms of this virus. Thank you.
Okay, thank you for your presentation. Now is Q&A session. Judges, you may ask your question now.
SG, please.
Okay. I would like to ask, where did you get this idea from? How come you have this idea to study this particular topic?
Okay, so I think we've all been affected some way by the SARS-CoV-2 pandemic. And then the thing is, we were doing a bit of reading because we were very curious. We wanted to know more about this pandemic that's affecting all of us. Mm-hmm. So we were reading up about what people have found out, what proteins interact with what from the virus. And the thing is, we noticed that in one of the papers, they did mention that severe cases of SARS-CoV-2 infected had higher levels of open reading frame 10. And the thing is, it was only noticed in severe cases. And we found this very particular and a bit interesting. So we decided to do a bit of research on it, and hence this project came about.
Okay, very good. So, sorry, yeah, continue on, please.
Sorry, to add on, we also had the help from our mentor to help us and taught us how to actually design our own plasmid, as can be seen from the slide. So we designed the plasmid constructs and decided on what type of restriction enzyme sites to design and insert in.
Because it seems to me that this kind of work actually is quite at a higher level. So I don't know where that's why I'm curious about, say, where information you get and what kind of technique you get before you start your study. Any help from, say, from NUS or from other institutes that help you to this project?
From NUS.
Yes, so we worked with a mentor. So when we saw this research paper and saw its interesting result, we actually proposed the idea to our research mentor, and he also agreed that it was a very interesting find. So then we started to have many ideas about and many hypotheses on like a potential project. So it kind of became this.
Okay, thank you. Professor Chen, please.
Thank you. So to begin with, I'd like to know more about ORF10, because it seems to be the target or the subject of your study. So do you know where does ORF10 express in the virus, or where is it located in the viral particle? Is it in the surface?
Open reading frame 10 is not a structural protein, so it's one of the NSFs, so non-structural proteins that come within the viral envelope. It is 144 base pair long RNA, or also is 38 amino acids long.
Yeah, I understand that. So how, if that's the case, how would you explain ORF10 could interact with cellular proteins if it's not a structural protein?
So it is accessory protein that comes within the virus, and it has already been known and established that viruses use their own proteins to interact with host proteins to help them replicate themselves and evade the host immune system. And hence we already suspected that the virus has its own proteins that interact with proteins within the human cell. And hence we did this investigation to find out which proteins specifically interact.
Okay, so one last question is about, you mentioned the signal peptide in the human interacting proteins of OFS10. So do you have any idea about why a signal peptide containing proteins is responsible for interacting with ORF10?
We first found the human peptide from E2 hybrid. So we use E2 hybrid to screen through hundreds of thousands of colonies, and we use split ubiquitin to find the potential interactions of which region ORF10 actually interacts with.
Yeah, so why signal peptide? Do you have any explanation to that, or just a coincidence?
Okay, so it makes sense to us that it would interact with a part of MHC class 1 so that it would continuously be able to replicate itself. And as for signal peptide, it is usually would be removed when B2M is added in or moved in, or in a sense put a part of MHC class 1, and then it will bring the viral antigens to the cell surface membrane to be presented to the immune system. So the idea is that if it prevents the signal, it removes the interacts with the signal peptide and then causes degradation, it will then prevent MHC class 1 from fully forming, and thus the cell is unable to display its viral antigens.
Okay, thank you very much.
Okay, thank you. Thank you, judges. Thank you, team. Team, you may leave the meeting room now. See you tomorrow again in the activities and award ceremony. Okay, take care.
Okay, thank you. We're looking forward to it.
Bye.
Thank you.
Bye.
Bye. Hello. Can you unmute? Yeah, good, great. Do you have the verb member? Yes.
Yeah.
Hello, hello. Would you unmute your microphone?
Yes.
Yes, that's great. Hello, hello team. Hello. So I would prefer team you unmute your microphone anytime during your presentation, starting from now. Okay, don't mute it starting from now. Okay. So I have to make sure all of you, your microphone now is ready. That's great. Okay, welcome to Global Youth Science and Technology Bowl 2021. I'm Haley, the MC of this judging panel. This team is from Thailand. Okay, first we would like to thank you for attending the judging online today and for your effort made in the past few months for developing the project. Today we are glad to have two judges, Professor Edwin Chen, the Chinese University of Hong Kong, and Professor Zhang Xiujin from City University of Hong Kong to be judges of the competition. Please note that my window will turn into a countdown timer when the judging officially starts. You will have 5 minutes to present your project when sharing your PowerPoint. We will remind you when the time is up and you have to stop the presentation. There will be 4 minutes for the Q&A session after your presentation. Do you have any questions? Great. Okay, you may now share your presentation material. And I'm now setting the countdown timer for you. So, are you ready to start?
Yes.
Okay, start please.
Hello, we are from IABC 10, food deodorant stickers made from silver nanoparticles. As a teenager, we are facing a lot of problems about food odor. It may not be a health problem, but people who have these problems may lose their confidence and cause bad personalities. Food odor mainly occur from bacteria that react with sweat. Our feet can produce up to 1 liter per week, since it's easy to have bad smell. So, we are interested to develop the sticker sheet that can inhibit microorganisms and deodorize food odor efficiently from through membrane and celluloid bacteria. Nanoparticles are particles which are tiny in scale. They will have more space to contact to microorganisms. And our objective is to develop the silver nanoparticle sticker sheet from through membrane and celluloid bacteria, and to study physical properties and ability to inhibit microorganisms that cause food odor from the silver nanoparticle sticker sheet.
Next is our methodology. Our project has 4 methods. First is bacterial cellulose separation. We grow the bacterial cellulose from Acetobacter salinum and ground it into a powder. Next is separation of sticker paper. We have 3 ratio of joint membrane is to bacterial cellulose ratio, dry and flat, and then test with Turing Hester to test what solution has the best Trier strength. Third is separation of silver nanoparticles from hydrogen pills. After synthesis silver nanoparticle solution using hydrogen pill extract, we need to identify silver nanoparticles by using UV spectroscopy, TEM, MSc, and MBC to make sure that we can really extract the silver nanoparticles from hydrogen pill extract. Next is development of sticker paper. The sticker paper was coated with silver nanoparticle solution and coated with glucomannan as a water repellent, and test the sticker sheet with Turing Hester and multi-day abrasion and peeling tester. And the last method is bacterial cellulose efficacy test of silver nanoparticle sticker sheet and study of the effectiveness of sticker developed to inhibit microbes in artificial sweat. Next is the result discussion. First is our sticker sheet. Combining the membrane with bacterial cellulose, we have found that the formula 3, which has the ratio 1:1 ratio, has the highest Trier strength. Then the hydrogen pill extract, an orange color solution, turned it into a dark brown color solution, after adding silver nitrate solution. Next, we have found that the 10 millimolar silver nitrate has the highest absorbance value and highest silver nanoparticles yield. The MIC in S aureus and M centaureus is 11.83, and MBC in more bacteria is more than 11.83. This is our SM images. After analysis with EDS technique, the weight percent of silver element is 4.55%, and the yellow color in this EDS layer image indicates the silver elements. And after coating the sticker sheet with glucomannan, it decreasing the abrasion resistance of the sticker sheet and has a 0.03% weight loss of silver nanoparticles, and also has a higher trace value than the coated one. The stamp head count method showed that the developed sticker had more than 99% efficacy of inhibiting bacteria. The distribution method showed that the developed sticker had clean zone on its interiors up to 30 mm and its areas is 20 mm. Eventually, the result of studying the effectiveness of sticker develops to inhibit microbes in artificial sweat. Our sticker can maintain a wide clean zone after spraying artificial sweat 8 times and then rapidly decrease to the end. And next topic is design of invention. We have the initial model and the new model to make it smaller, more modern, and attractive design.
This is our innovation, food deodorant stickers using silver nanoparticles. Our innovation has the potential to kill bacteria using silver nanoparticles that acts as an antibacterial substance. Moreover, our product is coated with glucomannan that allow water resistance. Subsequently, our product is tested for tearing strength and resistance through material abrasion test and peeling tester. In a nutshell, our product can be used up to 7 days without being teared and still effective. These are the steps in using our products. First, peel off the plastics that wraps around our products. Then stick the stickers in the forefoot region of the insole. Then insert the insole back into the shoes. Last but not least, our product is environmental friendly because it can be recycled into 3 easy steps. First, dissolve the used stickers in water and the silver nanoparticles will turn into silver ions. Then reuse the silver ion solution and synthesize the silver nanoparticles again. Combining with the jute membrane and cellulose bacteria, we'll get new food deodorant stickers. Thank you for listening.
Thank you, team, for a presentation. Now, judges may ask you some questions. Okay, Professor Chen or Professor Chen, please.
Okay, I'll start first. Okay, very interesting invention. So first of all, I would like to ask in particular, is your silver nanoparticles specific for the 2 bacterial strains that you mentioned only, or it is a general strategy to encapsulate or to target bacteria in general? If so, then what would be the criteria for the nanoparticles for selecting bacterial strains?
Oh, well, the silver nanoparticles has many range of size and shape. So we need to identify that our silver nanoparticles is okay for our bacteria or not. So we have the analysis with TEM. We have found that our silver nanoparticles has a range from 10 to 45 nanometers, and it is okay to use with our bacteria. Sorry, you unmute.
So the bacteria were killed after being targeted by the nanoparticles, or they were just immobilized?
They were killed.
Yes.
Because they have like mechanism in like destroying the membranes and react with the enzymes inside the bacteria.
Okay, great, thank you.
Can you unmute?
Okay, sorry. I would like to ask, in your study, you used 2 bacterial strain, the Micrococcus and Staphylococcus. In fact, are these 2 bacterial strain responsible for the odors in our feet?
I think it depends on like the age as well, like, and different regions. I think like different bacteria in the food. Yeah.
Because even though you confirm that your nanoparticles can kill the bacteria, if those bacteria are not the ones who cause the odor, it doesn't mean that your product is effective, right? So how did you test the effectiveness of your stickers in terms of the odors, right, generated? Did you test say that the odors by, I don't know how you can test, right? You ask someone to smell it or whatever. Have you tried that to test your effectiveness of your stickers?
Well, we have we didn't test yet because it has many complicated.
We have to do the consent. So like we only test with like the experiments, but not in human. Yeah. But it will be our future plan to do it.
Okay. Okay. Another question is, you mentioned it can be used for 7 days, right?
Yes.
Yeah.
So,
based on the bacterial count or what criteria did you use?
Well, we infer from our last experiment, which is the study of the effectiveness of the silver nanoparticle sticker sheet. Yes. And we have found that after sprayed artificial sweat with bacteria 8 times, that means that we can use continuously 7 days without being tear and still effective. Yeah.
Okay. Thank you.
Thank you, judges. Thank you, team. You may now leave the meeting room and see you tomorrow again in the activities and award ceremony. Okay? Take care. Bye-bye.
Bye-bye.
Yes, great. Hello, team from Thailand, right? Thailand. Hello, welcome to Global Youth Science and Technology Bowl 2021. I'm Haley, the MC of this judging panel. First, we would like to thank you for attending the judging online today and for your effort made in the past few months to developing the project. Today, we are glad to have Professor Edwin Chan from the Chinese University of Hong Kong and Professor Zhang Xiujin from City University of Hong Kong to be judges of the competition. Please note that my window will turn into a countdown timer when the judging officially starts. You will have 5 minutes to present your project with sharing your PowerPoint. We will remind you after 5 minutes and you have to stop the presentation, okay? There will be 4 minutes for Q&A session after your presentation. Do you have any questions?
No.
Oh, great. Okay, please share your presentation materials now. And I'm going to set the timer for you. Okay, are you ready to start?
Can you see it?
Yes. Okay, please start now.
Can you see the razor?
Yes, we can see your share screen now. You may start now. The time is counting down.
Good morning, honorable judges, ladies and gentlemen. Welcome to our presentation entitled Investigation of the Title Palm Umbrella for More Than Unique Position. We are from Stola Video School. My name is Untitled Chapter. My name is Untitled Chapter. Living in a tropical country like Thailand with hot and rainy season, the umbrella is an important need, but the normal umbrella has some problems. For example, it's susceptible to the growth of mold easily, its color will fade easily, it's not completely prevent bleeding, and finally, it's not to die. To solve this problem, we have come up with a project, and specifically we study the effect of hydrogen on the intensity of indigo dye. We study the effect of hydrogen and phenolphthalein on the color intensity and fairness of indigo dye. We study the effect of hydrogen and phenolphthalein on the color on the durability of indigo dye fabric, and we design and manufacture of hydrogen umbrella. So we suppose and put it to the experiment. For the first, we compare indigo fabric with hydrogen and indigo fabric without hydrogen by using a colorimeter to measure LAB value, and we found that indigo fabric with hydrogen have a better color intensity. Then we go to compare a different ratio of hydrogen, and we found that 0.6 grams of hydrogen are proper to use that to produce a palm umbrella because 0.6 and 0.8 have written different values.
Part 2, 2.1. In this part, we compare the color intensity of indigo fabric decorated with hydrogen and indigo fabric decorated with hydrogen and zinc. And from the experiment, indigo fabric that decorated with hydrogen and zinc is more intensity than indigo fabric decorated with hydrogen only. And so the next part, we compare the color intensity by different ratio of zinc oxide solution. And we use zinc oxide 0.8 gram because zinc oxide 0.8 gram and 1 gram have a little different color different value. Part 3, 1. In this part, we expose indigo fabric to light for 10 days and compare the different color intensity before and after dyeing. And we found indigo fabric decorated with hydrogen and zinc is best for prevention color fading after light. And 2, we wash indigo fabric 10 times and compare the different color intensity before and after washing. And we found indigo fabric decorated with hydrogen and zinc is best for prevention color fading after washing sand. In the next 2 points is main point of project. First, compare resistance against mold. This part, we mix agar, mold food, put indigo and observe the goal of mold. And we can know indigo fabric decorated with hydrogen and zinc is best for prevention mold. It can prevent 92.3%, but normal indigo can prevent just 43.1%. And the second, compare UV protection. We measure UV on the fabric and bottom of the fabric. And we found indigo fabric decorated with hydrogen and zinc is best for prevention UV. It can prevent 83%. But normal indigo can prevent just 40%. First, we hit indigo fabric level 1, 2, 3, And we found indigo fabric level 3 is best for waterproofing. Conclusion part 3. Indigo fabric decorated with calcine and zinc is best for prevention UV and mold resistant. Normal indigo fabric, if soaked with wax, it can waterproof, but it's made melting in room temperature of a tropical country like Thailand. But if we soak with calcine, zinc, and wax, it's very well because when mixed with wax and zinc oxide, it will be has higher melt point.
From the experiment, we found that indigo fabric with calcine and zinc oxide are proper to use to produce a calcium umbrella. So we are going to design and get it to invent a calcium umbrella. The conclusion. So our innovation is a better durability to mold resistance, power setting, and UV protection. It is friendly to the environment. It can decrease equipment costs and increase the value of local product. It can solve the local problem, and finally, local could get more income. Thank you. So this is our umbrella.
Thank you. Thank you for your presentation. Now, time for judges to ask you some questions. Judges, please start now. Professor Chen or Professor Chang?
Okay, maybe I start first then. It's interesting study. We know chitosan can absorb pigment, right? That's well known because some people use chitosan in absorbing dyes in wastewater treatment. And so it's a good idea to use it on umbrella. But have you tried how long last? Although you show some study, the color fading, but is there any restriction on the materials that can be used in umbrella for this kind of chitosan zinc oxide complex? You know what I mean? Is there any requirement for the materials used for the umbrella? Say what kind of cloth or polymer that can be used, right? In your study, the materials of the umbrella, is there any restriction or limitation? Because different kind of cloth may pick up the dye differently.
It can be used in every situation in the daily life, but sometimes it have slightly heavier. But if you have more beautiful and with good property, yes.
Okay. Professor Chen, do you have any question?
Okay. It's very interesting project and the product is beautiful. I want to buy one. So regarding the UV protection, since this is an investigation project, do you have an explanation to how the chitosan and zinc oxide mixture can absorb the UV light? In one of your slides, you mentioned UV protection. Is it correct? Yes, yes. So how do you measure that?
I measure that by using UV mixer.
We use UV meter and measure UV on the fabric and bottom of the fabric and compare the difference.
Yeah. Okay. Yeah. So obviously there's a very clear difference between normal and chitosan plus zinc oxide, but can you speculate the reason why? Why chitosan plus zinc oxide has the UV protective effect?
Because the zinc oxide can prevent UV.
And calcine prevent UV same.
Okay. Right. Okay. Thank you.
Yeah.
Thank you.
Okay,
Okay.
thank you, thank you.
Thank you. So, thank you, team. You may leave the team right now. Sorry, you may leave the meeting room now. See you tomorrow again in the activities and award ceremony. Okay, see you tomorrow. You may leave meeting room now. Bye.
Yes, we can. Hello.
Great. Hello, team from Indonesia, IABC 13. Okay, welcome to Global Youth Science and Technology Bowl 2021. I am Haley, the MC of this judging panel. This is team from Indonesia. Okay, first we would like to thank you for attending the judging online today and for effort made in the past few months for developing the project. Today we are glad to have Professor Edwin Chen from the Chinese University of Hong Kong and Professor Zhang Xiujin from City University of Hong Kong to be judges of the competition. Please note that my window will turn into a countdown timer when the judging officially starts. You will have 5 minutes to present your project with screen sharing of your PowerPoint. You have to stop the presentation when the time's up. And there will be 4 minutes for the Q&A session after your presentation. Okay, do you have any questions?
No.
Great. Okay, now please share your presentation materials. And I'm going to set the countdown timer for you.
Can you see?
Yes. Great. Okay. Are you ready to start?
Yes, we are ready.
Yes. Okay. Start now.
In 2019, Indonesia was awarded the title of the top dirtiest countries in the world. Not only that, Indonesia also occupies the 5th position with the title the dirtiest river in the world. If we as the nation generation do not make change, who else will change the title of Indonesia in the eyes of the world? Assalamualaikum warahmatullahi wabarakatuh. Hello everyone, let us introduce ourselves. My name is Ratna Kawandari.
And I'm Amalia Putri.
We are from
Islamic State Senior High School 1 of Lamongan.
Here we would like to present our innovation with the title
Natron Natural Environment Detergent, Innovation of Natural Detergent from Belimbing Hulu and Coral Leaves. According from the Indonesian Commercial Newsletter, total detergent consumption for the territory of Indonesia in 2010 reached 449,100 tons and is estimated to continue to increase in line with the increase in population in Indonesia every year. This causes pollution such as in Kabiti Maruda River, DKI Jakarta, Kali Item, North Jakarta, and also several rivers in Lamongan area. This incident occurred because detergent sold in the market contain hazardous chemicals that are difficult to decipher, such as surfactant alkyl benzyl sulfonate (ABS) and linear alkyl sulfonate (LAS, so that they can pollute the river flow.
Belimbing Hulu or Averhoa bilimbiel is a plant that grows a lot in the Lamongan area and is usually as a cooking ingredients by the community. The compounds contained in this fruit are saponin, which come from foam so that they can be used in the detergent industry. Not only Belimbing Hulu, Kelor plants or Moringa oleifera population in Indonesia is also very large. Kelor is a common plant in rural areas where the leaves are never used at all. The compounds in Kelor leaves are protease enzymes that can bind to proteins.
From this background, we are innovating to make an environmentally friendly detergent, and the result of this research is Natron, which is a natural detergent made from Belimbing Hulu and coral leaves. From this background, we decide 3 problem statements. The first, how do we make a natural detergent with the combination extracts of Belimbing Hulu and coral leaves in removing stain? Second, what is the best concentration of natural detergent with the combination extracts of Belimbing Hulu and coral leaves in removing stain? And the third, how is the effect of natural detergent with the combination extracts of Belimbing Hulu and coral leaves in removing stain?
In this research, we do some steps to make natural detergent from Belimbing Hulu and coral leaves. The first, observation and sorting coral leaves and Belimbing Hulu, and then wash both of them with water flow. After that, drying coral leaves only and start with extraction process of Belimbing Hulu and coral leaves. After extraction both of them, mix Belimbing Hulu extract and coral leaf extract according to several samples. After that, we did the detergency test and the last is packaging process.
There are some of the samples that we have designed. We make into an sample like this table, with A is Belimbing Hulu extracts and B is coral leaf extracts. After make some sample, we do some tests. The first is detergent foam test, with the best sample is sample 9, and the second test is detergency test, with the best sample to remove stain is sample 5, that all stain are gone. Because sample 9 is only have foam much, but it cannot remove stain like sample 5, we choose that the best sample is sample 5 with a moderate amount of foam but can last a long time. And there are image of detergency test. We can see that sample 5 is cleaner than another sample. And this is the result of the saponification test that we have conducted at the laboratory of the Faculty of Pharmacy at Erlangga University, Surabaya. This calculation is calculated using the saponification value formula. This average value 10.7 with the average value of 4.5%. The saponification value is a sufficient value, so it is suitable as neutral detergent.
This is for your designs for Natron. In 2021, test Natron in laboratory. In 2022, legality and product development. In 2023, branding and marketing expansion. And in 2024, community empowerment and mass production.
Conclusion. The best concentration of Natron with a sample 5, which consisted of 50% Belimbing Hulu extracts and 50% coral leaf extracts. With the saponification value, the average value is 10.7, with the average value of 4.5%. Sample 5 can remove stain on the clothes.
That's all from our team presentation. We believe that our innovation is useful for our environment. Thank you very much for your nice attention. Netron, time gun, but environmental hygiene never done. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you, team. Now you have 4 minutes to have the Q&A sessions. Judges, you may start to ask questions.
Okay, maybe I start first. Can you hear me?
Yes, we can.
Good. Okay. In your study, you test the effectiveness of the detergent using the 2 criteria, right? The foam they produced and also the stain they remove, right?
Right.
So how did you quantify the amount of foam and the amount of stain removed? Quantitatively, I mean, how do you quantify?
Can you repeat again?
How did you quantify the foam? Because you use foam, right, as a criterion for assessing the effectiveness of the detergent. The amount of foam, how did you quantify that?
It might be 3 centimeter. So the foam can be the foam from our detergent. The hanger is 3 centimeter.
3 centimeter.
Yes.
Okay. You measure the height of the foam.
Yes.
Okay. And how about the stain? You say it can remove the stain. So how can you assess how much stain is removed?
How much stain that can
be removed?
Yes. For stain, the first is soy sauce. The second is sauce. The third is oil, and the fourth is soil.
But how did you quantify the amount of stain removed? You say, okay, it's all removed. What does it mean by totally removed? By your eyesight or by any instrument to measure whether there's any stain left behind? How did you assess that? You know my question?
Can you repeat again?
Because you say, okay, the detergent can remove the stain effectively, right?
Right.
But how did you draw that conclusion? How did you measure whether there's any stain left behind? Because there may be a color change, a color intensity change.
This is we test our product.
Yeah.
Before we use Netron, and this is after we use Netron, and the best sample is sample 5. So this is very effective to wash clothes.
Okay.
You can see.
Okay, that's good. Okay, Professor Chen, please. Okay.
I would like to know about the active ingredients of your Netrons.
Active ingredients in our products have 2 active ingredients. The first is saponin. The saponin is the natural surfactant. So saponin can replace chemical surfactant in the detergent. And the second is protease enzyme. The protease enzyme is one of the phosphate compounds. In detergent, phosphate compounds usually use chemical ingredients. So we replace chemical ingredients with protease enzyme.
Yeah, so have you determined the percentage of the saponin and the proteases in your Netron products?
All Netron products?
Yeah.
Saponification?
How much does the saponin and the proteases are present in your product?
In care leaves, protease enzyme is 4.27 units per milligram, and in saponification, in saponin in belimbing wulu, in 1 liter of extract belimbing wulu, 75% is saponin.
Okay, so how did you measure these? How do you determine the percentage and the amount of the ingredients in your product?
We
Did you do experiment to determine the amount and the percentage of the active ingredients?
active ingredients we know from exciting projects, we know from exciting experiment, so we can conclude that our product have some ingredient, has some percentage, and we also test neutron in laboratory.
Oh.
And the result is 10.7. 10.7 is a sufficient value for detergent. So it can remove stain and also it's safe for environment.
Okay, thank you very much.
Okay.
Thank you.
Thank you. Thank you. Thank you, judges. Thank you, team. You may leave the meeting room now. Okay. See you tomorrow again in the activity and our ceremony. Okay. Take care.
Thank you very much.
Thank you. Bye bye. Can you all unmute your microphone?
We are in the same place, so we're using.
I see. Okay, that's good. That's fine. Okay. Welcome to Global Youth Science and Technology Bowl 2021. I'm Haley, the MC of this judging panel. This team from Vietnam. Okay. First, we'd like to thank you for attending the judging online today and for your effort made in the past few months for developing the project. Today, we are glad to have Professor Evelyn Chen from the Chinese University of Hong Kong and Professor Zhang Xiujin from City University of Hong Kong to be judges of the competition. And please note that my window will turn into a countdown timer when the judging officially starts. You have 5 minutes to present your project with sharing screen, or you have to stop the presentation when the time's up, okay? And there will be 4 minutes for the Q&A session after your presentation. Do you have any questions?
No.
Okay, so you may now share your PowerPoint, and I'm going to set the countdown timer for you.
Okay, so can you see?
Yes.
We will start now.
Okay. We can see the your PowerPoint. Are you ready to start?
Yes.
Yes.
Okay. Start now.
Ladies and gentlemen, today we are Group IBBC 47 from Vietnam, and we are going to talk about our experiments that can help to dissolve the polluted wastewater in Tolic River using titanium dioxide.
Well, Tolic River is one of the main rivers of Hanoi, and it used to be a clean and wonderful place. But sadly, over the years, people have been dumping all sorts of garbage, varying from antibiotics, food waste, to all the stubborn inorganic waste, like plastic bags. And naturally, plastic items take from about 1,000 years, and even our daily plastic bags take from 10 to 20 years to naturally decompose. And in contrast to that number, approximately 197,000 tons of plastics are discharged into Hanoi rivers every year, including the Tolic River. And that's why we are offering our beloved city a solution by using titanium dioxide. Titanium dioxide is a photocatalyst which has an absorption mechanism and can be used to break up carbon chains of inorganic matters and make them become water and CO2.
So now I'm going to demonstrate the process of preparing titanium nanoparticles in the laboratory. So the method that was used during the process is called Hydrothermal Technique. In the first step, we created a mixture of titanium dioxide and isopropanol. And then the mixture was slowly dropped into 900 milliliters of water and 50 milliliters of isopropanol. The resulting solution was a colloid containing titanium dioxide nanoparticles. The next step is to centrifuge the colloid and let it dry at 120 degrees Celsius to get a white powder as a result. In the last step, the white powder was calcined at 400 degrees Celsius for 2 hours and obtained the final product of titanium dioxide nanoparticles.
Next, we're going to talk about the results of the experiment. Xrd and SEM together give the exact knowledge about the particle size and the characteristics of the titanium dioxide sample. Firstly, this is the Xrd pattern of titanium dioxide at 400 Celsius degree. The anodized space was identified at 2 theta values, corresponds to the crystal planes according to the standard JCPDS. No characteristic peaks were detected for rutile phase, Brookite phase, or any impurities. Therefore, we can conclude from this result that we have created titanium dioxide. Secondly, the SEM can characterize the shape and the size of the titanium dioxide nanoparticles. The surface morphology of nanoporous titanium dioxide was characterized by SEM analysis as shown in the image. The pure titanium dioxide nanoparticles exhibited spherical morphology with clumped distributions, and the size of the particles is about 10 nanometers.
And then we will talk about the absorption spectrum. And a material absorption spectrum is a fraction of incident radiations absorbed by the materials of a range of frequencies. The absorption spectrum is primarily determined by the atomic and molecular compositions of the materials. And also, GBV spectrum is known as a molecular absorption spectrum. And the second one is a photocatalyst. And its definitions is a material which absorbs light to bring it to the higher energy levels and provide the energy to a reacting substance to make a chemical reaction occur. And when photocatalyst titanium dioxide TiO2 absorbs the ultraviolet radiations, or we can call that the UV radiations from sunlight or illuminated light source, it will produce pairs of electrons and holes.
Well, in conclusions, the titanium dioxide is excellent for the purification of Polluted River, and our team has successfully researched and synthesized the titanium dioxide. The Xrd diffractions help us to identify the appearances of titanium dioxide, while the SEM characterizes the shapes and size of the nanoparticles. The titanium dioxide photocatalyst has been found to be more effective than any other antibacterial agents, and it also shows a long-term effects. Therefore, our team will do more research to apply the titanium dioxide to more aspects of life. Thank you for listening.
Okay, thank you for your presentation. It's Q&A session. Judges, you may start asking questions. Professor Chen or Professor Chen?
Thank
Professor
you
Chen,
for the
please.
presentation. May I ask if titanium dioxide been used in a similar application before?
Yes, it has been used for the purification of the Polluted River for the from the Japanese groups and companies. So they offer us the option of using this this method of water purification, but unfortunately, the Vietnam government denied it because we haven't. We have some problems.
Yeah. So the other thing is how would you compare your product with other existing strategies to treat polluted wastewater?
Oh, okay. So normally people usually collect the trash and the garbages in the rivers and then either bury them underground or burn them, and both have harmful effects, such as when you bury them, then deforestation happens, and when you burn them, then harmful gases and ashes will be released into the air. But our method is to decompose them, and after a while they will become just water and CO2, which are way less harmful than those existing methods that we are currently using in our country.
Yeah, I see. So is there any nanoparticle approach that had been applied before your titanium dioxide one? And if so, then how is it compared to what you have produced? Is it better than theirs?
In our case, applying to the specific Polluted River, then no nanoparticle methods were ever applied.
Well,
and that explains why the river is so smelly and black. We're trying to solve that by using the new method.
Because the titanium dioxide can dissolve in methylene blue, which is the hardest substance to decompose. So we can conclude that the titanium dioxide is safe for the environment also.
Yeah. And moreover, the titanium dioxide is a cheap material with stable chemical properties, and also titanium dioxide is very durable.
Okay, thank you very much.
Okay, thank you. Now you mentioned at the beginning, one of the problems of your river is the pollution by plastic waste, right?
Yeah.
Yes.
But how, can titanium oxide handle plastic waste?
Yeah, it can. It's just wait, what?
Can we have just a second to discuss?
I mean, plastic waste is a global problem.
Yes.
And one reason is because it's very difficult to degrade plastics. They are very stable. So can titanium oxide handle plastic waste?
Nhưng mà thầy bảo là nó biết đang có chỗ CH được.
Vào
Plastic là các chỗ CH nhưng mà decompose tại sao? Plastic là các chỗ CH nữa.
So team, do you have answers to Professor Jen's questions?
Can we discuss about it a little bit?
Okay, it may leave it to your further discussion after this judging time. Okay, so because our judging time is finished and over. Okay, so thank you judges, thank you team, and you may leave the judging room now. See you tomorrow again in the activities and award ceremony. Take care. Bye bye.
Okay, thank you.
Thank you very much. Have a good day.
Have a nice day.
Have a good day. Hello, good morning. Good morning. Welcome to Global Youth Science and Technology Bowl 2021. I'm Haley, the MC of this judging panel. This is team from Indonesia, right? First, we would like to thank you for attending the judging online today and for your effort made in the past few months for developing the project. Today, we are glad to have Professor Edwin Chan from the Chinese University of Hong Kong and Professor Zhang Xiujin from City University of Hong Kong to be the judges of the competition. Please note that my window will turn into a countdown timer when the judging officially starts. You will have 5 minutes to present your project with screen sharing or any other materials. You have to stop the presentation when the time's up. And there will be 4 minutes for the Q&A session after your presentation. Do you have any questions? Any questions?
No.
No? Oh, sorry, I cannot hear you. Can you speak again or speak louder?
No, miss, that was very clear.
Yeah, okay. Now it's very clear. So, do you have PowerPoint to share?
Sorry?
Do you have PowerPoint materials to share?
Yes, we are sharing the screen right now.
Yeah, you may share your screen now.
Okay, can you see our screen?
Yes, we can see clearly. Okay. Oh, are you ready to start?
Yes.
Okay, please start now.
Ladies and gentlemen, one of the ways to prevent coronavirus and to boost our immune is to eat nutritious food. A lot of people likes eating catfishes, as you can see from this picture. A lot of catfish farmers does not support healthy nutrition and does not using hygiene for the catfishes. So we as consumers consume the fish and get the disadvantages such as diarrhea and cancer. So from this major issue, we have decided to take action and create an innovation by title, Passing Pellets, catfish feed to support healthy nutrition. My name is Rafael.
My name is Rafli.
And I'm Uma, and we are from
Avicenna Cinere High School, Indonesia.
Ladies and gentlemen, before we get on with the presentation itself, I would like to direct your attention towards the originality sheet on the left, which has already been signed by our school's principal, indicating that our innovation is original and has never been done by any party before. So the outline issue that we are facing is actually omnivorous fishes, especially freshwater fishes, which are being fed unsanitary things. But the thing is that these fishes range in different countries, but in Indonesia, the fish that's put into focus is catfishes, which is why our project focus is towards the catfishes. The catfishes that are raised in Asia, some of the to give it less sanitary feed, such as animal innards and feces, which will lessen the quality of the product during harvest and can cause multiple diseases such as diarrhea, cancer, and other digestive diseases, which is why it is very needed for the availability of a cheap pricing feed that's fulfilling for the catfishes.
Most of catfish farmers rely on factory-made pellets when it comes to catfish feed, but some of them use alternatives sometimes. One of the most common alternatives are papaya leaves. This is because papaya leaves contain carbohydrate, fat, calories, calcium, and protein that can fulfill the catfish daily needs. In addition, papayas are also seasonal organic waste. We use papaya leaf which has been utilized by the society maximally to take a part in the organic waste problem. And then the next ingredient is rice waste. Because our purpose is to reduce organic waste, we use rice waste as well, considering the nutritional components in the rice as well. And then the last ingredient is fish meal, which is very easy to find and usually sold for a very cheap price. Fish meal also acts as the binder in the dough.
So how we were able to combine all the ingredients to make our pellets? Let's watch this video together. So first, we need to prepare the ingredients, this being 2 parts papaya leaves, 2 parts rice, 3 parts fish meal, and 1 part water. Next, add all the ingredients into a bowl and stir until all the ingredients are incorporated. This mixture will then be blended to smooth out the texture of the dough, which resemble the texture of cookie dough. After that, we can start shaping the dough into small balls around 0.5 to 1 centimeter in diameter. After putting the wet pellets under the sun for 2 days, they will harden and finally can be used for catfish feed.
Ladies and gentlemen, we didn't only come up with the ratio for making pasipong pellets, but we also observed the effects that the pellets have on live catfishes. And just to shorten the explanation, the catfishes that were given pasipong pellets underwent a more significant growth and seemed to be more agile compared to the catfishes that were given pre-made pellets.
Ladies and gentlemen, in conclusion, catfish are important in food sources and fishery, but catfishes are also given less than suitable feed in its cultivation. That's why pasipong pellets is one of the solution for this problem. And then While at it, we also use organic waste that in our products to reduce organic
Okay,
waste.
team, time's
That will be all
up.
for our presentation. Thank you so much for your attention.
Thank you. It's time for judges to ask you some questions. Okay, Professor Chen or Professor Chen? Professor Zhe, okay, good.
Okay.
First John, please.
Okay. I noticed that you're in your table showing the red bucket and the green bucket, right? So
Yes.
So how many fish did you use for each treatment group?
Well, to use a full-scale farm-like simulation, I guess, or test would take a lot of effort. But so we went with like 5 catfishes in one bucket and 5 in another bucket with the same density as a real pond would. And we made sure that the age and the size of the catfishes were the same and we gave them the same dosage as regular farmers would with regular catfish feed.
Okay.
Versus the passive method.
You use the rice in say feeding the fish, right?
Yes, it was one of the ingredients in the catfish pellet.
So why did you use rice? Is this a carbohydrate source to the fish, or you have any other reasons?
Okay, so I will explain the nutrition content of each ingredient. So first, we have the papaya leaves. So papaya leaves contains a lot of good proteins, vitamins, carbohydrates, and carbon dioxide. And to boost the nutrition content of the papaya leaves, we use rice because rice contains a lot of good carbohydrates for the catfishes. And for the 2 other ingredients, last ingredients, and that is fish meal and water. It's mainly for the texture control. So if the dough is too runny, we can add more fish meal. And if the dough is too solid, we can add more water. And in addition, fish meal contains a lot of good proteins. So it can boost the nutrition content of the dough itself.
Okay, thank you. Professor Chen.
Okay, thank you, Mr. Chang. I am curious about why you need to mix the 3 ingredients together. I mean, is there any chemistry or chemical reactions that would happen between the papaya leaves, rice, and fish meal? Have you tried like separate them and then use them separated as a food source for the fish? Would that make a difference when compared to the mixture?
Okay, thank you so much for your question. Well, in catfish farming and cultivation, papaya leaves are actually used as alternative feed. So they're used as them all the time because they're not nutritionally. And the other ingredients that we use, like rice, and are actually the ingredients that would help support papaya leaves so that the pellet become a main pellet rather than an alternative pellet. So if it's about a chemical reaction, we're not entirely sure there is one. Looking for the nutritional components.
So the one last question is about the duration of the feeding. Okay, it's a short period of time. So how would you be able to draw a conclusion with a rather short period of treatment period to demonstrate the effect of this phenomena? Because all these fish I can see are like adult fish, so they grew up by feeding on standard food, right? Before they were treated with the pellets that you made. So how would you control that?
Actually, the fishes that we used in the sample were actually younger fishes. They're not adult fishes. So it's very possible if a fish, a catfish from the from its hatching, but rather from it's allowed to eat pellets. they could start pellets, that would be fine. Maybe we can alter, since the size, since there's like, we can maybe alter the size of the pellets to better suit smaller fishes. And here my friend is just about to show you what they look like.
Okay, right, it's very nice. Thank you very much.
Okay, thank you.
Thank you, judges. Thank you, team. You may now leave the meeting room. I'll see you tomorrow again in the activities and award ceremony. Take care. Bye bye.
Thank you so much for
Thank
your
you so
attention.
much, judges.
Thank you. Bowl 2020. Would you please unmute your microphone?
Yes.
Okay, great. I'm Haley, the MC of this judging panel. This is team from Indonesia. First, we would like to thank you for attending the judging online today and for your great effort made in the past few months for developing the project. Today, we are glad to have Professor Edwin Chan from the Chinese University of Hong Kong and Professor Zhang Xiujin, City University of Hong Kong, to be judges of the competition. Please note that my window will turn into a countdown timer when the judging officially starts. You have 5 minutes to present your project. After 5 minutes, you have to stop the presentation. And there will be 4 minutes for the Q&A session after your presentation. Do you have any questions?
No, that's clear enough. Thank you.
Great. Okay, please share your presentation materials now. And I'm going to set the countdown timer. Okay, good. I can see your PowerPoint. Are you ready to start? Team, are you ready? Would you please unmute your microphone?
Yes. Okay.
Yes,
Okay, ready to start now, right? So please start. Please
The
unmute
research
your
of
microphone.
is held. Ladies and gentlemen, respected judges, in the current pandemic situation, this quote proves to hold even more veracity than in the 2000 years it's been around. Today, we present the innovation we originated as an act of people to achieve this work during the present state of the world.
A naturally colorful health enhancer from the colorfully vibrant butterfly pea flowers, purple yams, and Augusta flowers. Labiri pudding. Hang on, where's okay. Sorry, we had some technical problems.
We wish you all a very good morning. My name is Vinesha. My name is Dinda.
I am Nisrina, and we are from Akisana Cinere Senior High School, Indonesia.
Ladies and gentlemen, how often do people check up on what their snacks are made of? With the vast amount of processed foods today, it's no longer surprising to find artificial substances in the snacks we consume every day.
Erythrocin, aspartame, monosodium glutamates. We can find additional artificial substances easily these days. And sure, they might not be vehemently harmful, but does that mean that they're healthy?
Now, more than ever, it's not the time to take change with our health. However, seeing as to how well-liked snacking is, the total restriction of snacks will be improbable. Which is why we provide a pertinent alternative that could satisfy the desire to snack while being able to enhance the consumer's immune system.
Labiri pudding aims to take the attention of consumers with its unique ingredients and properties to prevent the risk of increasing health concerns in the community.
We did this by utilizing natural resources that are abound in availability and health benefits. In that, we resulted on butterfly pea flowers, red Augusta flowers, and purple yams, all of which were then composed into a pudding to appeal to the public as a popular snack form. By extracting the ingredients into a liquid and combining them with agar-agar powder, labiri pudding by combining By extracting the ingredients into a liquid and combining them with agar-agar powder, labiri pudding took on a visual appeal of being expressively colorful while still being safe and healthy for the body.
Numerous health beneficial food compounds are included in this pudding, from the general vitamins such as vitamin A and C to those such as selenium and phytochemical substances. The liquid extracts used in labiri bring many beneficial effects. What more, our choice of ingredients also enabled us to achieve such uniquely vibrant colors with only the addition of an acid or base such as the common lemon or baking soda.
This coloring opportunity was due to the anthocyanin contained in butterfly pea flowers, Augusta flowers, and purple yams that act as acid-base indicators. With this, it was possible to consume health. Instead, we were able to change the hues by simply combining it with a squeeze of lemon or a small sprinkle of baking soda. Of course, labiri was proven beforehand by a selected group of examiners as an assurance for its consumers.
Of the total amount of testers, 80% reported back with claims of feeling more energized or refreshed. The remaining reported back as neutral with no filings of complaints about discomforts in taste or general negativity caused by the consumption of labiri. With that, we can then safely announce that labiri pudding is safe for consumption.
From all that, we see that labiri pudding strength lies in its ingredients that enable this pudding to have health beneficial effect, which additionally can attract consumers with its naturally gained vibrant colors. However, after further evaluation, it would appear to be even more enjoyable to the consumers if the flavor present in labiri could be more pronounced and varied. In future developments, this could be a possible focus point to work on.
That being said, labiri pudding already has prominent economic value in the market. A uniquely aesthetic, healthy snack thrives in today's marketplace and could be used as a good business opportunity. And with that, we conclude our presentation. Thank you so much for your time and attention.
Okay, thank you, team. So that's it for our presentation, right? Finished. Okay, good. Now it's time for our judges to ask you some questions. So Professor Chen or Professor Jen, please.
Hey, thank you very much for presenting this project to us. So you mentioned in the title of your project that the product that you have made is a health enhancer. And also from the presentation, you mentioned that there are several claimed effects after the consumption of 12 months of the pudding. Was that correct?
Yes. So that was the effects a week after consuming that pudding.
Yeah. So what's the frequency of the intake of the pudding during the 12 months period? Is it once a day, or what's the portion and the frequency?
Sorry, the period is just a week, so 7 days.
A week.
So
okay.
yeah, it was said in the slide, but you probably missed it. It's
Right,
okay.
right.
So in the week, we gave them 2 puddings. So every 3 days, they would have it once a day, every 3 days, and that was the claimed effects that they reported back to us.
Okay, so how did you determine such a frequency and the amount of intake?
So when we did our literature, our research, we figured out that pudding, or this pudding is made of agar-agar powder. It only takes 3 days to be fully digested into the body. So we were basing it on that. And because we only had a certain amount of time, because in the pandemic, we only chose to have it twice. So that was why it was in a week. And that was why there was 2.
I see. And also another question is about safety. So you also mentioned that this is a safe product. So how did you measure the safety of the consumption?
Yes, because we
So this product is safe because we only use natural ingredients. For example, the 3 main ingredients are butterfly pea flowers, Swetagasta flowers, and purple yams, all of which are naturally gained. And for the agar-agar powder is made out of the red walls, the cell walls of red seaweed, which is also natural. And for any additional substances that we put in, they were also safe and commonly used in everyday situations, such as sugar, baking soda, and lemon. They are all safe to eat. And despite all that, we still did a test, which is the graph you see before in the slide, and none of them claim to have any side effects or any illnesses after consuming labadi pudding. From there, we can then safely announce that this pudding is safe for consumption.
Okay, thank you.
Thank you for your questions.
Okay, I have a question about your design. Okay, you mentioned that most of the people who have tried your product, they feel energized or refreshed, right? But have you done any blind tests? Then what I mean is, okay, whatever you offer to the people, they shouldn't know whether this is your pudding or other kind of pudding. Have you done any kind of this blind test?
Yes, so we provided them with this. We didn't tell them that it was our pudding. We told them that it was a pudding, and we asked them to tell us how they feel after consuming this pudding in a certain amount of time. And we also gave it the general pudding from the market, such as strawberry pudding or chocolate pudding, to compare the effects.
That means they didn't know what kind of pudding actually they took, right? Am I right?
Yeah, we just said this is the pudding made out of Radagasta flowers, and this is the pudding made out of with the chocolate flavor. And we asked them the comparison of the effects a week after.
That means they knew about what kind of pudding they took.
They knew what kind it was, but they didn't know that it was supposed to be a health beneficial pudding.
Okay. And just the final question. The ingredients you use, you mentioned they are natural. Okay.
Yes.
But we know naturally doesn't mean that it's safe, right? So what's your comment on that?
So yeah, that is a good point. We have done our literature and our research on this. So we use agrarian resources such as flowers and yams that are safe for consumption and that have been consumed for many years actually, but they're not very well known by the by the public people right now. So they were traditionally used, but because it has been a long time and people tend to prefer, more modern stuff like oranges or apples, we try to bring this back into attention and to say that we have all these resources that we could be using, that we could be utilizing, and we should be making use of it.
Okay, thank you.
Thank you so much for your question.
Thank you, judges. Thank you, team, for your participation. You may leave the meeting room now. See you tomorrow again in the activities and award ceremony. Okay, take care. Bye-bye.
Thank you so much.
Hello, team. Would you please turn on your camera and unmute your microphone?
Yes.
Okay. Okay, great. Hello, welcome to the Global Youth Science and Technology Bowl 2021. I'm Haley, the MC of this judging panel. First, we would like to thank you for attending the judging online today and for your effort made in the past few months for developing the project. Today, we are glad to have Professor Edwin Chan from the Chinese University of Hong Kong and Professor Zhang Xiujin from City University of Hong Kong to be judges of the competition. Please note that my window will turn into a countdown timer when the judging officially starts. You have 5 minutes to present your project with your PowerPoint sharing. When time's up, you have to stop the presentation. And there will be 4 minutes for the Q&A session after your presentation. Do you have any questions? Great. Okay, now please share your presentation materials. Okay, we can see your PowerPoint now. Are you ready to start? Okay, please start now.
Good morning, everyone. I'm Anna Wu.
I'm Sam Lo.
I'm Bo Ban.
The work introduced to you today is converting newspaper into soil for blossoms. We would like to see if we can develop a new type of artificial soil which can be used in arid areas to retain sufficient water for farming. What is the reason for us to conduct this research? It is because global warming has been posing threats to our food production around the world. For example, in the United States, more than 90% of the land is dry in winter, which causes a low crop yield. There are various types of artificial soil. However, most of them are mixed with compost, ash, manure, and many more ingredients which may pose harmful effects on the human body. Therefore, an artificial soil which could maintain the natural properties of soil has been developed in this project. Our project aims at developing an artificial soil which is environmentally friendly, non-toxic, convenient, and has good water retention.
For the materials, we have put newspaper, polyvinyl alcohol, and chemical fertilizer together in our artificial soil. We chose these materials because PUA shows strong water-absorbing property. Calcium-containing fertilizer has been added to the materials, assumed that this can act as a cross-linking agent for PUA. And we also hypothesized that coarse and nanocrystal cellulose in newspaper can be extracted by acidic hydrolysis to increase the strength and water absorption properties of the PUA paper pulp. For the methods, paper pulp was first made by digesting blended paper in Europe's sulfuric acid and use calcium carbonate being neutral, then converted into soil bed by phase inversion technique.
9 samples were prepared for the optimization of different materials. It is found that the higher proportion of fertilizer, the higher viscosity and integrity the sample is. So we assume that PUA can cause linking with this fertilizer because we have found that the samples without adding any fertilizer could not turn into gel and remain in liquid form. So we assume that it has successfully proved that fertilizer could properly act as an effective cross-linking agent for PVA paper pulp into gel to further convert the hydrogel into solid state for convenient transportation and usage. We have used phase inversion. We have found that only samples of 1:1 ratio 1 could instantly transform to solid bits, so that this ratio was continued to be used in the following tests.
3 samples show the best water retention were medium contents of fertilizer with pulp to PVA ratio of 1:1, 1:3, and 1:5. Throughout the course of the test, they are all higher than the natural soil. For the rest to know is whether our artificial soil can be used in an arid environment. Limited amounts of water was added to the crops, and prolonged shining of lamp was used to stimulate the arid condition. The application test showed that 100% of artificial soil resulted in the fastest growth for 24 days. Trisom grew up to 6 cm in the simulated drought environment. 2/3 of artificial soil mixture followed up to 4.5 cm, whereas other samples did not germinate at all. Chinese cabbage also showed the same results of growing in the application test.
The cost of our artificial soil is $2.3 per kg.
As a conclusion, considering the necessity and effectiveness, artificial soil made from non-toxic ingredients, including used paper, biocompatible PVA, chemical fertilizer has its great potential in farming in arid environments. The recycling of used paper could also have elevated the problems arose from the landfill waste. This is the end of our presentation. Thank you.
Thank you. Now, judges will have some questions for you. So, Professor Chen or Professor Zhang, please.
Okay, maybe I start first. You in your study, you add chemical fertilizers into your artificial soil. So how long lasting is the chemical fertilizer? Will they leach out from your product?
Our chemical fertilizer is we use it into our soil by the cross-linking agent. Since we found some information about the other country, for example, we found some information which called the calcium, and it's very good for the plants to live. So we put it in our
our soil to let the pH constantly and let the plants grow fast.
Yeah, conclude, the fertilizer contain calcium cations that can use for the cross-linking agent. And often we use chemical fertilizer in farming, so we add it into our project.
So it can reduce the leaching rate, right? The leaching rate of the chemical fertilizer, am I right? Because the chemical fertilizer, they are water soluble, they can leach out quickly. So
we use as our cross-linking agents and quickly cross-linking into hydrogel, so it can be released when you use it.
Okay.
And when you.
Okay, good. And another question: when you compare your results with the natural soil, because we know different plants, they need different types of natural soil. What does it mean by natural soil, right? Because it depends on the percentage of different sizes of sediment and organic matter, right? We have some soil which is more porous, right, with more air space. Other soil actually with less space. Is the soil, the natural soil you use, good for the plants you use in this study, say choy sum and Chinese cabbage? Is that natural soil really is the best type of soil for this kind of plants?
Yes, because our compared our comparison is that is focused on the aerated area, and the choy sum and the Chinese cabbage need lots of water to let it grow. So we use choy sum and Chinese cabbage for the compare things, so it can more so our soil can very suitable for the arid environment.
Also, you can see that 75% of artificial soil add to the natural soil. We mix natural soil with our artificial soil, and this can see that our artificial soil is better than natural soil. And is more suitable for vegetable to grow. We compare that how suitable is our artificial soil compared with natural soil, but natural soil doesn't fit this cabbage and choy sum. So we can see that our artificial soil is better than natural soil.
Okay, thank you.
Okay, thank you, judges. Thank you, team. You may leave the meeting room now. See you tomorrow and again in the activities and award ceremony. Take care. Bye-bye.
Bye-bye.